I'd like to introduce Dr. Monica Embers, who is a professor in the Division of Bacteriology and Parasitology at the Tulane National Primate Research Institute, where she studies the pathogenesis of Lyme disease spirochete Borrelia. Her research investigates the natural course of Borrelia infection in non-human primates to gain a better understanding of the clinical aspects of Lyme disease in humans. So please welcome Dr. Monica Embers.
All right, thank you. Let's see. Let's see if this advances. Yes. All right. Well, thank you for the kind invitation to return to focus on Lyme. I always love coming to Arizona, and I get very thirsty coming from Louisiana. So it's really a joy to be here and to be able to talk to you today about some of our very, very recent findings. In fact, they're somewhat incomplete, but I think we're getting a good picture of what's happening. And so I just want to start off by talking about the fact that in both reservoir and incidental hosts like humans or non-human primates, persistence is the norm for this pathogen. It's not the exception. And so these bacteria have devised really complicated, and they've evolved over time to become immunoevasive, what we call immunoevasive. So they know how to evade the immune response and persist. And so we've learned a great deal about what happens in mice that are infected with Borrelia burgdorferi. And we're starting to learn a little bit more about what happens in humans. And in nature, mice are a reservoir host. That means that they are there to propagate the infection. The ticks feed on the mice, pick up the bacteria, and transmit it. And so there's a difference between that and the incidental hosts, like humans or non-human primates, because mice don't really get sick. And the level of spirochetes in their body are much higher. So we want to learn more about what happens in primates. Okay, I already talked about those. So when I first entered this field, it was because I was so intrigued by how pathogens evade host immune responses, and Borrelia burgdorferi does it better than any pathogen. It evades the host immune response at so many different levels. And this can actually be split into two components. One is the ability to evade the immune response, that is to avoid recognition. And the other is to actively suppress the immune response. And so we know that these pathogens do both of these things quite effectively. And so what I'm going to focus on today is the component of the active immune suppression. So this is just one really good example of how Borrelia can suppress the immune response. And so Borrelia induces the expression of this cytokine called IL-10. And this is a broadly immunosuppressive cytokine. So it prevents other cells from making other chemical mediators that recruit an immune response. And it does it both, it affects both macrophages and dendritic cells. And so these two cell types are sort of the link between the primary infection and the development of an adaptive immune response, because they present antigen and they tell T cells and B cells that, hey, it's time to start targeting this pathogen. And so Borrelia is interrupting this at that phase quite effectively. And we know these are studies in mice, but we know that this happens with human peripheral blood mononuclear cells that are stimulated with Borrelia burgdorferi. They do make IL-10 as well. So not only does the pathogen suppress the immune response, but the ticks, when they feed, their salivary components prevent the immune response from happening. And this happens both at the level of the innate immune response and at the adaptive immune response. I'm going to start pointing, so I need to just get... Okay. Can you hear me? Is this on? Okay. So here we have These are T-cell inhibitors, and also we have B-cell inhibitors. So these are the two big arms of the immune response that are induced by invading pathogens. And we know that tick saliva on its own can prevent B cells from becoming activated. So in my mind, what's happening with Lyme disease is that you have three components. First, you have the infection that you have to deal with, and that's just the pathogen in the body existing and causing problems. Then you have inflammation, and inflammation is typically what causes the pathology in the disease, causes the disease in the tissues. And you can have chronic inflammation if you don't eradicate that pathogen. And then you have the suppression or the alteration of the immune response. So all those three components are working together to create this disease. And what I think needs to happen is we need to be able to very rapidly diagnose and eradicate the pathogen, because even remnants of the pathogen could potentially result in chronic inflammation. And we don't know how long the immune suppression lasts and how that can affect the co-infections or subsequent infections. So a couple of years ago, there was a paper that came out of the Baumgarth lab, and she was here last year and talked about this. And they were able to identify something very important about Borrelia infection. And so what they did was this experiment to determine the suppression of humoral immunity to an unrelated antigen. So how does Borrelia infection affect a response to something else? And so she took the flu vaccine, a flu vaccine, and gave it to mice that were either infected or not with Borrelia burgdorferi. And this just shows that they added transgenic T cells specific for a peptide of the flu antigen. So this is a way that they can measure specific T cell responses. And so after they infected the mice and vaccinated them, then they monitored the immune responses over time. And what they found is that they did get the production of antibodies these are antibody secreting cells, and these are different isotypes of antibodies. And they were fairly similar between the two groups. And then they looked at the T cells to the transgenic T cells specific for the antigen, and they found that there was a bit of a decrease in those that were infected with Borrelia, but it was not enough to be significant. So here is the big take-home message here from this study. And so what you're seeing here are the antibody responses to the flu vaccine in mice that were not given Borrelia and mice that were given Borrelia, infected with Borrelia. And you can see that the antibody responses were dramatically reduced in mice that received, that were infected. And this went on into the bone marrow. They looked 9 weeks post-infection and found that there was a significant drop in the percentage of antibody secreting cells. So then you have to think about functionality. So the next thing they did was they took immune serum from vaccinated mice that were either infected or not infected, and they injected that into naive mice and infected them with the flu. So the question is are the antibodies produced protective? And they measured this by looking at body weight, whether or not the mice got sick. And so the mice that received the serum from the uninfected vaccinated mice were fine, and the mice that received the serum from the vaccinated infected mice did not do well. So there were a number of conclusions that came from this paper. And one of them, they spent a lot of time looking at germinal center responses, and these are the interactions in the lymph nodes between the T cells and B cells and how they generate specific responses. And these were basically skewed. They happened later than they should, and they fell apart pretty quickly. And so we know that some of the responses to Borrelia antigens are dependent on T cells and some are not. And it turned out that this mechanism of suppressing the immune response is independent of the T versus B cell antigens. And here's the key. Borrelia infection suppresses the induction of long-term protective immunity to a co-administered vaccine antigen. And so herein they identified this as a very important aspect of Borrelia infection. So what about humans? What do we know about humans? I think the picture that we're starting to see with respect to the functionality of the immune response to an infection is also influencing antibiotic treatment. So we treat most patients with doxycycline. This is a microbiostatic antibiotic. It doesn't kill the bacteria. It relies on a good immune response to clear the infection. So what happens if you don't generate a good immune response? Well, this was a study that was published out of Bill Robinson's lab at Stanford. And what you can see here is these are plasmablasts. These are and this is the percentage of their antibody response to 5 different antigens, Borrelia antigens, at the height of their response. And these are the patients who have post-treatment Lyme disease syndrome. And we compared this to a Lyme arthritis patient that was positive for all 5 of them. And so that's where we got the percentages, just as a comparative value. And so what we found is that 9 of the 10 patients that returned to health generated really strong IgG. This is antibody, a particular antibody type. That's the most specific antibody, IgG, to 2 or more antigens in our Bioplex assay. And all of the patients who ended up having post-treatment Lyme disease syndrome had either weak responses to all of the antigens or produced a response to this one antigen, OPPA2, which we've seen when we've tested chronic patients. We see a response to that antigen. And so we think that, you know, this is indicative of how patients are responding, that you need to have a good antibody response to begin with in order to have an effective treatment. And so this doesn't really apply to those patients who have gone for a long period of time without getting treated, because we know that that becomes a lot more difficult. These are only patients treated in the early phase. So the explanations are that host differences exist in the ability to generate humoral responses or antibody responses. And there are also strain differences among Borrelia and co-infections that can influence the immune response. It could be that the antibody responses target antigens other than those in our assay. We don't think that's very likely because we've done Western blots to confirm our findings, and that some post-treatment Lyme patients had EM but were not infected with Borrelia. And I don't think that's very likely either. So we set out to look at how Borrelia infection affects the immune response to an unrelated antigen in non-human primates. And so why would we use non-human primates for this research? First of all, we know that rhesus macaques most closely mimic Lyme disease in humans. All the different signs and symptoms that you see in humans, you can see in macaques, things like erythema migrans, carditis, arthritis, and importantly, neuropathy of the peripheral and central nervous systems. Mice don't get infection of the brain or the peripheral nerves. And they also don't get rashes. And we also know that the spirochete burden in tissues following dissemination is very small, like it is in humans. It's very hard to recover these spirochetes from infected humans or primates. And that's one of our challenge, a constant challenge of ours. So we know that compared to mice, the disease course is more similar to that of humans, and compared to human samples, we know the infection history, and we can also look at post-mortem tissues to find the spirochetes. So we hypothesized that infection with Borrelia burgdorferi would non-specifically impair B-cell responses. And so we had 9 4- to 5-year-old male rhesus macaques in the study, and they were split into 3 groups with 3 monkeys per group, those that were infected, those that were immunized and infected, and those that were immunized only. And so we chose to use this HIV-derived GP130 antigen as our immunogen because I'm at a primate center that mostly studies HIV AIDS, okay? And so this is really well characterized at our center. And we infected the monkeys by tick bite. So this is what the experimental outline looked like. We infected the monkeys, we immunized them. 1 month later, we gave them a boost, and then 1 month after that, we gave them another boost. And this was actually, for those of you who are really into the science, this is a recombinant varicella virus encoded GP130 that we gave for the primary in the boost, and then we gave the subunit for the second boost. And so after this through this experimental outline, we looked at antibody levels both to IgM and IgG, and at B and T cell populations, both from the whole blood and from the lymph nodes, in terms of phenotype, functionality, and specificity. So let me walk you through this. These are the antibody responses generated to the HIV antigen in monkeys that were vaccinated only and those that were vaccinated along with the Borrelia infection. And so you would expect that you would start to see something after the first boost, which is at 2 months, and then 3 months is after the second boost. And these are this is actually a concentration of antibodies, so it's quantitative. And so this monkey here, KC09, generated an early response. KC97 and KP73 generated a low response at 2 months and a good response at 3 months. This is exactly what we would expect to see. Then when you look at the monkeys that were vaccinated in the presence of a Borrelia infection, you see one monkey, this one had an early response in the pre-immune, so I think it maybe had some pre-exposure to something that could cross-react, but steadily inclined with every boost. The monkey in red, he generated a good response at 2 months and then a good response at 3 months. And this guy, LF45 in the blue, didn't do anything. A little bit of a response at 2 months, but the boost had no effect. So this is one case in which we're seeing what Nicole Baumgar saw in the mice, where the presence of the infection prevented the generation of antibody responses. Now, we work with monkeys, so we have a hard time establishing statistical significance with an N of 3, but this looks like a case in which we do see failure to generate a response. And so then we also looked at the response to Borrelia antigens, and what you can see here is that you have each group here is a monkey that was immunized over the period of time. And each bar that you see is the antibody response to one of our 5 different antigens. So there should be a set of 5 bars in each set. So you would expect that the immunized-only monkeys don't have any response because they didn't get infected with Borrelia. We have, let's see. These monkeys generated really good responses. This one did 2. This one did 2. This is our guy who didn't generate a GP130 response, and he produced antibodies only to this OPPA2 protein. And then we have another animal that didn't convert to. We have 2 other animals that didn't seroconvert to Borrelia, and you could say, well, they're not infected, right? We had at least 9 infected ticks feed on all of these monkeys. Interestingly, this one here, KL37, after we did necropsy, we put all their tissues in culture to see if we could recover Borrelia. This almost never works. I wish we could grow Borrelia from monkey tissue cultures, but we can't, historically. We've been in the process of developing a new culture method, that's another story. And lo and behold, from this monkey that was completely seronegative, we had lots of positive cultures. In the lung, in the brain, in several regions of the brain, it was positive. So here we have another case where we have an infected seronegative monkey. So that's another case we can study. And I'm very excited because we're seeing things that you see in the human population in our non-human primates. And what's great about this is that we give them all the same bacteria. So whatever we see is a difference in the host, right? Host variability. Okay, am I going over on time? Okay. So this is the, now we're getting deeper into the immunology here. And so we take the blood cells from the periphery, they're called PBMCs, or peripheral blood mononuclear cells, and we stain them with antibodies to find different markers that tell us what kind of subsets they are. So this is the flow cytometry gating pattern that we use. We have, these are non-switched memory cells, these are switched memory cells, these are double negatives, these are naive, and then we sort them out based on what kind of expression markers they have. Okay, so what you're looking at here is the frequency of the percentage in the B cell populations of this particular type. So here, these are all just B cells. So each line is a monkey, as you can see here, and it looks like they all have about the same percentage of B cells. Then when you look at the memory cells, what we're looking at specifically are differences between the immunized only, the infected immunized, and the infected only in terms of how the populations change over time. So this is going to be day 14. That's going to be when you start to see responses develop after the primary boost. Day 70, this is after your second boost, so you would expect to see elevations. So in the immunized monkeys, you can see that with the memory cell generation that you do see a spike in memory cells. And you do see a similar thing a similar trend in the infected immunized monkeys, except for this blue line, that's our LD59. That's our guy that didn't generate responses to GP130. So keep an eye on this blue line in the middle. And then in the affected only, we see pretty good memory cell generation. Okay, so these are B1 cells. These are effectively sort of non-specific B cells that are part of the innate response, so the antibodies they produce are not as, they don't have as high affinity or avidity for the pathogens. And so in the immunized only, you see a peak here at day 14, which you'd expect, and then you start to see them come up. Infected and immunized, we're seeing the same trend, but it's definitely decreased in amplitude. And then in the infected only, again, we're seeing about the same trend. These are the IgG producing cells. So these are the antibody producing cells that are generated. Immunized only, for some reason this monkey didn't generate very good IgG, but this is the trend you would expect to see with an increase in a peak in day 70. If you look at the infected and immunized, 2 of the monkeys did, but look at our guy here. Nothing, just really didn't increase in IgG producing cells. And then in the infected only, there really aren't a whole lot of increases in IgG producing cells after the infection. So this is kind of what we expect with Borrelia infection anyway. These are IgM producing cells, and CD21 is a maturation marker, so these are effectively IgM producing cells that have matured and those that have not matured, and you would expect to see like a mirror image. So these are the immunized only, and these are the infected immunized. So where there's a drop here, you should see an increase here. Where's an increase here, you see a drop here, because they're replacing, these cells are replacing these cells effectively. And we don't see huge differences here. And these are the plasmablasts. So these are the switch memory plasmablasts. So these are the activated antibody secreting cells. And you see pretty good responses in the immunized only, and a peak early in the infected immunized, but you want to see a peak a little bit later like this one. And for LD59, that didn't happen. And then the infected only really didn't generate good plasmablasts at all with Borrelia infection. Let's skip over that. All right. And then again, we're looking at IgM producing cells, and the infected, these are the switch memory mature. Here's what, I mean, after the generation of IgM, you would expect to see a switch to IgG production in a normal infection or immunization, and we see that with this, we see that with these monkeys, but here you see an increase in IgM producing cells, especially with this this LD59. And so this is really atypical. And most people know that you see persistent IgM in Borrelia infection. And look, you're seeing increases in IgM producing cells in this case as well. And for those of you who don't know, I mean, IgM is the early produced antibodies that are not as specific, and then they switch after a period of maturation and encounter with antigen, then they switch to IgG, and those IgG antibodies are a lot more specific and for the pathogen or the antigen. All right, so now we're switching over to T cells. So what are the T cells doing? And let me remind you, this is actually just peripheral blood mononuclear cells. We haven't even gotten to the lymph nodes yet. T cells are either central memory or effector memory based on their functionality. And what we would expect is to see an increase in effector and memory, or central memory and effector T cells with immunization and boost, which we see pretty decently here. There's our guy not doing a whole lot, LD59. And then, so we don't see huge differences there. But then when we looked at activated T cells, Ki67 here is a marker for activated T cells. And you see that early on upon the immunization in these animals. But look at the infected and immunized. You don't get that early peak in activated T cells. And in the infected only, you just have it sort of delayed. A sort of delayed response, which is kind of what we would expect based on the mouse studies. So we still need to analyze all the IgM antibodies. We still have lots of lymph nodes to look at. We've done the flow cytometry. We've also done activation of T cells from the lymph nodes to determine if they're functional, if they're making interferon gamma in response to the GP130 antigen. And so we still have a lot more to add to this story. But just to conclude, one of our 3 infected animals failed to generate an appreciable antibody response to the vaccine antigen. It appears that IgM-producing cells are more frequent and persistent in the infected primates. Clearly, our memory B cells and plasma blasts are reduced in population in those animals that were infected with Borrelia burgdorferi. And whereas CD T cell memory populations appears similar, activation of T cells was somewhat dampened in the Borrelia burgdorferi infected primates. And so I could not do any of this without the support of my lab. Most of them are shown here. In this case, Dale Martin and Amanda Tardo and Nicole Hasenkamp contributed. Mary Jacobs runs our tick colony, and our vet. And then in the Division of Comparative Pathology, Bobby Pehar, he's the immunologist that sort of walks me through all the data. I don't know that I'm a card-carrying immunologist, which might be evident. And I want to express my most sincere gratitude to the Bill and Marion Cook Foundation for the funding for this project. And finally, I promised Liz Horn that I would say something about the tissue biobank, because a lot of times I have patients contacting me that want to contribute or have different cases, and if there are any samples that could be contributed. Recently, I was contacted by somebody who had tumors that wanted to have them analyzed, and they should be deposited. They should register with NDRI and then be deposited in the biobank. So, thank you.